everyone, and welcome back to another installment of Teach Me in 10, a video series that's brought to you by LabTube, which is part of the Technology Networks group. And here at Teach Me in 10, we invite scientists to talk to us about their research areas or a scientific concept in less than 10 minutes, with the aim being to make science much more accessible. So my name's Lucy, and I'm a scientific content producer here at Technology Networks. And today I'll be speaking with Audrey Bergeron, an application scientist, and Hilary Sherman, a senior application scientist from Corning, which is one of the world's leading innovators in material science. They join me today to speak about 3D cell cultures. Welcome to Teach Me in 10, Audrey and Hilary. We're delighted to have you with us for our question and answer video. So to kick off, Audrey, could you please tell us what is 3D culture and why does it matter?
Yeah, so traditionally adherent cells are been cultured in 2D. So think more like a monolayer, they're flat, they're against something like a plastic or a coated plastic. And in 3D culture, instead the cells are grown more like in aggregates or clumps. So those cells in 3D culture are experiencing a different environment compared to 2D. They're getting more cell-to-cell interactions. When they're grown in a clump like that, you end up with these gradients where you get oxygen or nutrients, more of that for the cells on the outside, whereas the cells in the middle of that clump are experiencing a lot less of that. And that's important because if you think of something like a solid tumor in your body, that's going to be more like that type of mass or clump, and the same thing with an organ. So when people are performing toxicity studies trying to find drugs in order to kill cancerous cells, when they're testing them on cells that are grown flat in a monolayer, those cells all see the same concentration of that drug. Whereas if you're working with something that's in 3D, then you're going to also be looking for a drug that can penetrate that mass and reach those cells in the middle. And then if you're looking at something that's more like trying to mimic an organ, something like a liver, for example, it's been shown that hepatocytes, so your liver cells, when they're grown in 3D, they actually live longer and maintain function for longer than when they're just grown in 2D.
Hilary, where is 3D cell culture having the most impact? In your research areas or drug discovery, or maybe in personalized medicine? What are your thoughts?
Yeah, so as Audrey mentioned, cancer is a prime example of where three-dimensional cell culture really adds value. As she mentioned, tumors in the body are not two-dimensional, they're three-dimensional. So being able to study them in an environment that's much more similar to how they would be in the body, has really helped to push, cancer research. Additionally, we're starting to see a lot of interest in personalized medicine with the ability to grow organoids in matrices such as Corning's Matrigel matrix. Here, personalized medicine is really having a lot of interest because you can grow a patient's biopsy to study their particular cancer. Or we've also seen great progress in the research area of cystic fibrosis by being able to grow intestinal organoids in three-dimensional culture and thus studying their response to different drugs to correct the cystic fibrosis mutation.
So then, Audrey, what are the different 3D models and how do you know what model to use and which tools are best?
Yeah, so it really depends on what your application is, what your goal is with your 3D model. So 3D models are typically made either using scaffold-based or scaffold-free methods. In a scaffold-based method, you can get something like some type of matrix that's keeping the cells together. In a scaffold-free method, typically their cells are grown more like in a suspension. At Corning, we offer several different platforms depending on what your end goal is. So for example, if you just want to be growing clumps of cells, spheroids, you're not looking for something very specific like trying to do organs replication. You're not doing something that complicated. We have our ultra low attachment platform. And so we offer that in a flat version and multi-well plates up through larger vessels. And that's more if you're just scaling up these 3D cell cultures, but it doesn't matter to you that they're all of the same size. However, for some people, they want to be doing more like a drug screen against 3D cell culture models like spheroids. In that case, it's important that each of those spheroids is the same size. So for that, we offer our spheroid microplates so that you end up with the same size spheroid in each well across the plate. For some testing, however, there's not enough cells in a spheroid. You might only have something like 5,000 cells. So you won't get a large enough signal. And so then we have our Alplasia product line where you can get many spheroids within one well to help amplify that signal. And then finally, there are more complicated models as well. or you might be trying to do something that's more like mimicking an organ, for example, and you're typically working with stem cell models, and this is with organoids. And for that, you need more biological information. So you need something like our matrigel in order to help give that information to the cells. And that's a scaffold-based method.
Perfect. And then Audrey, could you also describe what the typical assay readouts are for 3D cell cultures?
Yeah, so again, it depends on what your end goal is. For someone who's looking at drugs to try to target and kill cancer cells, you're doing some type of assay at the end to see which cells have been killed or how many cells were killed. And so that can be an ATP assay, or you can be doing live dead staining with that. And that's usually more high throughput. There are also a lot of researchers who use high content imaging where they might want to look at something more specific about how the cells are being affected. And that can be a little difficult because you're trying to get high resolution images of these 3D structures. And so you need to be able to image through these structures, and you're also getting a lot of data, but there have been a lot of advancements in that area in recent years to help people with that work. And then finally, you could also be looking at something like how marker expression has been affected or if it's maintained in your models. You can either dissociate your cells to then do flow cytometry with them, or you could be extracting RNA or DNA from those models.
And what do you think are the most important considerations for optimization in 3D culture, Hilary?
Yeah, good question. So there is a lot to consider when transitioning from more traditional two-dimensional culture to three-dimensional culture. It's much harder for reagents to penetrate a 3D structure compared to a 2D structure. So this can be in terms of lytic reagents for a homogeneous cell-based assay, or it could be in terms of staining of your cells or permeabilizing your cells in order to accomplish staining. So sometimes using stronger reagents, more concentrated reagents, or increasing incubation time might be necessary. As Audrey mentioned, light penetration into a 3D structure for imaging can also be more challenging. So using confocal microscopy in order to get better light penetration and clearer images throughout the structure can be really helpful. It's also really important to consider the size of the spheroid for what you're trying to model. Maybe you want something really large that's going to have more of a oxygen gradient or more of a necrotic core similar to a tumor. Or maybe you want a smaller structure that's going to have a more homogeneous culture throughout. So these are aspects to consider when you're optimizing your assay. And so deciding how many cells to seed and how long to culture is going to impact the overall size of the spheroid on the day of assay. Additionally, if you want to actually recover your cells from your 3D structure and use some type of dissociation solution, you might need a stronger dissociation solution than you would typically use in two-dimensional culture, or you might just need to incubate for longer or do more pipetting in order to break up those structures.
And then finally, because we're almost out of time, I'm wondering, Hilary, how is 3D culture being handled in automation environments?
Yeah, great question. So, for liquid handling with three-dimensional cultures compared to two-dimensional, it can be a little bit trickier. With two-dimensional culture, the cells are very safely attached to the bottom of the plate. So there's little risk of inadvertently aspirating away your cells or moving your cells when you add fresh media or fresh reagents. But with three-dimensional culture, the cells are not attached. And so you could inadvertently aspirate away your spheroids or disrupt them when you're adding fresh cell culture media or drug compounds. So automation really has had to advance itself in terms of how it manipulates these 3D structures by adjusting the velocity in which the liquid is added or removed or the force. Additionally, having instruments that can work at a particular Z height so that they can stay far enough away from the spheroid when doing the aspiration or when doing the addition. And a lot of specialized instruments have come out recently that are specialized to work with three-dimensional cultures compared to 2D. Additionally, we're finding more and more instrumentation that are capable of handling viscous solutions such as 3D matrices, such as Corning matrigel matrix.
Mm-hmm.
So either by having cooling mechanisms to keep the hydrogels nice and cold and fluid, or by using syringe-based systems that are better able to handle viscous solutions than more standard, regular tip-based systems.
Well, thank you so much, Hilary and Audrey. It was great having you join us today.
Thanks so much. It was fun.
Thanks for having us.
I quickly wanted to take this opportunity to thank Hilary and Audrey for joining me here on Teach Me in 10 today. We'll be back next week for another instalment of Teach Me in 10, but if you can't wait till then, you can head over to our hub page, which we've linked down below, which is where you can see all of the videos that we've published so far.